Nice. Thank you, Judy. Dr. Connor, how are you?
Good, thanks.
Good. We finally got our show started.
Yes.
We should do a blooper reel. Hey, look, I really do want to thank you for coming into the show today. I was hoping we might be able to start off with you giving us a little bit of background on how you got into the field of research that you're in, and where your interest in that area sort of lay, and then maybe we can start to then talk about some of the specific, really interesting topics around stem cells and that sort of thing that you're doing.
Yeah, sure. So I came from a science background. My dad's a engineer, and so science was always something. I was really passionate about science at school, particularly biology, and I have memories of my dad helping me do science experiments and just doing science experiments at home. Like I remember being given a crystal set to make a crystal radio for a birthday present, which, if you gave that to kids these days, they'd look at you strangely. So it sort of was in my background, and I headed off to university with the aim of actually doing sports science. So I used to row for New Zealand competitively, and I had decided that what I wanted to study at university was how to make people perform better, a high-performance sport. And I got to university, and the one science topic I was never very good at was physics, much to my dad's disappointment. And discovered that physiology and exercise physiology was very much about physics, and it was just like, oh my goodness, what have I got myself into here? And this was back before they actually had a sports science program. So I started off under my dad's general advice of keep your options open, which I always think is a great advice. I took a whole range of subjects, including some psychology papers, and so it was at stage 1 university, and I was taking general psych, and I remember opening up the textbook and reading about the human brain and just going, oh my goodness, this is incredible. And that was probably the first time I'd ever really actually thought about the human brain and what it did. And that really got me hooked. And so I then decided to shift my interest from sports science to what we'd call now neuroscience, but I was thinking about more along the psychology lines. I took some psych papers in stage 2 and still some physiology papers. And then in stage 3, I had to finish off my degree. And at stage 3 level at Auckland University, they offered pharmacology. And I was flicking through the calendar, and I thought I needed to make up 2 more papers. And I looked at it and I thought, learning about how drugs work. Okay, that sounds pretty interesting. I'll have a go at that. And that was where things really fell together for me. So it was the whole concept about how drugs work in the body, and so not pharmacy but pharmacology. So it's the science of the function of drugs, how they work, and developing new drugs for treatment, and then hook that in with neuroscience. And of course we have all of these neurological disorders of which we have very few effective treatments. And that really, I was really hooked on that idea. Majority of my friends then left to go to med school, and I decided that actually I didn't want to go to med school because my passion was in neuroscience, and all I could see from neurology was that I would be talking to people and saying to them, I'm afraid we don't have anything that we can actually help you with. And I wanted to be one of those people that could actually turn around and say, we've actually found something that can help you.
That's cool.
And so I set off and started doing a graduate degree at Auckland Uni in an area known as neuropharmacology, so basically identifying new drugs to treat neurological diseases. That just was exactly where I fit. That was my niche, and it was just really, really passionate about it. Finished my PhD, and I went over to Northwestern University in Chicago, where I worked on the field of gene therapy just when it was starting out. And so this was looking at gene therapy to deliver a factor known as glial derived neurotrophic factor, which is naturally found in your brain, and it helps the development of cells called dopamine cells, which are the cells that are lost in Parkinson's disease. And so...
Are they receptors of dopamine? Is that...
No,
are they produced?
So glial derived neurotrophic factor is like the food for dopamine neurons. So when your brain's developing, it needs glial derived neurotrophic factor, or GdNF as we call it, because it's a very long word. It needs GdNF to develop those dopamine neurons, make them mature. And then as your brain, in your mature brain, it helps protect and sort of continue to feed those dopamine cells. And there's a lot of evidence that in Parkinson's disease, if you apply some more GdNF, it helps protect and prevent the dopamine cells from dying off. And so the concept behind that was we were using this, at that time, novel therapy of gene delivery to deliver the GdNF into the brain so that it could actually stay around long term and protect those cells that were still remaining.
So the GdNF isn't then a drug that you deliver like orally.
No.
It's delivered as part of a gene.
Yes.
How does that work?
So we use what we call viral vectors. So viruses have this, as we're all very familiar with viruses now with COVID, viruses have this ability to go in and enter into your cell, enter into the nucleus, and then make more copies of itself and then spread forth and multiply, basically. What we do in gene therapy is you actually cut out pretty much 99% of the virus, the infecting part, the part that makes it replicate itself, and instead you put in what we call the gene of interest. So that would be, in this example, the gene to make the GdNF protein. And so the virus with this GdNF comes along and it actually goes into the cell. It doesn't replicate itself, but it starts pumping out GdNF. So basically the cell starts making its own GdNF, or the cells around them start making GdNF, and you get this really large increase in GdNF. So it's a protective mechanism. It's not going to bring cells back from the dead. So, unfortunately with Parkinson's disease, most people don't realise they have Parkinson's disease until they get the clinical symptoms, at which case you've already lost about 80% of your dopamine neurons. So we can't bring those back. What GdNF has the ability to do, hopefully, is to maintain the remaining population. So you're going to prolong the very early clinical symptoms and not have the patient progress further and further into degeneration. Yeah.
And what is the cause of Parkinson's?
We don't actually know the exact cause of Parkinson's. So 10% of the population have what's called a genetic form of Parkinson's. So we know there's a genetic mutation. But as I said, that's relatively rare. It's only 10%. The rest of the population with Parkinson's disease, we call it sporadic. There's no real known reason for them to have developed Parkinson's. We think, though, it will be a combination of genetic and environmental factors. And there's, of course, as there is with anything like this, there's always been a huge range of suspects for the environmental factors. Things like rotenone was going around as being a popular thing, other environmental toxins and chemicals that you find in the environment. But there's no one causative factor for people to develop Parkinson's disease. And of course, it is a disease of aging as well too. So some people are more susceptible. They might have been prone to exposed to something during childhood or adulthood, and eventually it all comes together, and it's the dopamine neurons that start to die off and they get Parkinson's disease.
Is it targeted? Like with people with Parkinson's, is it just the...
Just the dopamine neurons.
It's just the dopamine neurons that cause the Parkinson, but do they suffer other gross degenerative effects on their brain, or is it just...
Yeah, they do eventually. So, of course, all brain cells connect to each other.
Yeah.
Okay. And so the initial target is the dopamine cells in the near called the substantia nigra, which is involved in initiating purposeful movement and stability of movement. So that's where you see Parkinson's patients with a tremor. Mm-hmm. They've got a very slow, sort of very upright gait. Some of them have very little facial expression, so very rigid face. And they all have different symptoms as well too. So there's no, as the clinicians will say to us, there's no one true Parkinson's case. All the cases they see are different. Again, that's where it's difficult to treat patients, because the cause of their disease, the symptoms they suffer, it's all individual. And so what you're doing is you're trying to target that final aspect, which is the degeneration of those dopamine neurons. So what's caused that? Is there a key pathway that it all comes together in? And so can you find that? Yeah.
And so it's not something that you can solve by producing more dopamine in the brain. It's actually the cells that use the dopamine,
so
or is it?
That's what so Parkinson's disease is one of the diseases that we do have a few treatment strategies, and they work for what we call the clinical symptoms. So they work for treating the tremor, the rigidity, the initiation of movement, and that is basically replacing dopamine. So, and that's an oral, it's a tablet called L-Dopa. It's the precursor to making dopamine. The patient takes it, gets taken up by their brain, and the remaining cells make more dopamine. So you're getting a bigger boost from a fewer number of cells. But eventually over time, because this is a progressive disease, you're continually losing your dopamine neurons. So those treatments such as L-Dopa doesn't stop the degeneration. So eventually the L-Dopa doesn't work anymore because there isn't enough cells for the L-Dopa to work on. And there are some other medications downstream that you can combine, but eventually there's not a lot that the actual symptomatic treatments can actually do. So what people are looking for, like with gene therapy and delivering GdNF, is to protect and maintain that population, that when a patient walks in and they've got a tremor and the doctor says, I need to send you to a neurologist, and the neurologist says, well, you've got early stage Parkinson's disease, that you can stop that or slow it down significantly so that they are still being able to be effectively treated by L-Dopa. Their symptoms are just, okay, I've got a bit of a tremor, take a tablet, it goes away.
So it's a combination of things to try to sustain them where they are now.
Yeah.
Does the GdNF is really interesting. Does the mechanism of how it protects those dopamine neurons give an indication of what is damaging the neurons?
No, it doesn't for GdNF, because GdNF is really like, as I said, it's protecting. So that's its role. Yeah. So we just put more protective factor in. Yeah. It's like wrapping it up in bubble wrap, right? Okay. It's not going to the root of the cause of the disease. Yeah. And so there's a lot of interest now about a protein called alpha-synuclein. And alpha-synuclein becomes mutated and aggregates and forms in patients with the genetic form of Parkinson's disease. But alpha-synuclein goes and forms these big clumps of toxic protein in what we call in the sporadic Parkinson's disease as well. So what is that alpha-synuclein doing? It's toxic to the cells. It causes the cells to die. What's the key pathways that lead to this forming in patients with genetic Parkinson's and with sporadic Parkinson's? Can you target something to prevent that big clumping of alpha-synuclein from happening? Mm-hmm. Is there something that you can do to break it up? So people are looking at various therapies there.
The mechanics of the cell mechanics, I guess, are amazing. Just the fact that as a child, I sort of imagine cells, they're just these magical things. And
they are magical things.
Well, they are.
Yeah.
But stuff just happens. But then over time, and with the advent of YouTube and videos showing 3D representations of what's going on in cells, what's quite mind-blowing is that they are little machines. Mm-hmm. There are mechanical components in there doing jobs. And that to me sounds like it's a really hopeful state, because it means that, like as you say, we've got clumps of proteins forming around cells. I feel as though, there must be some way we can engineer some sort of mechanical thing to go and clean the cells.
Yeah, well, you'd think so, but of course it's never quite that simple, unfortunately. What you learn with neuroscience and in the brain, the brain is just the spectacular organ that we have that we still don't really know its full potential. It's an incredible organ. If you think about everything that it's doing without you thinking about it, I was listening to something the other day about learning to read and about phonics, and how that every time you read, even as an adult, you still go through the whole phonics thing. Just your brain does it so fast now that you don't recognise it. And I was thinking, wow, there's so much load on the system. Yeah. And then when one little thing goes wrong, you have quite a massive, effect. Cell mechanics, as you call it, which is a good term for it, or, how the cells process. One little thing goes wrong there, a whole nother pathway of stuff happens. And that's where, as you're saying, it's like, oh, we can just go and let's just get rid of the protein. And, you're not the first one to say that, because there's a huge billions and billions of dollars of research that have gone into just getting rid of that alpha-synuclein protein. And, there's some companies with some, they've put products out into clinical trial now looking at exactly that have spent billions of dollars looking at that, and we still don't know whether it will actually be effective.
Or what the impacts might be.
Exactly.
The side effects of it.
Yeah. So, it's as you said, it's like, oh, we'll just mop it up. Yeah.
How do we just mop it up?
20 years later, we're still mopping it up.
We're trying to figure out what we can build to do that. Yeah. But I think it's interesting being able to just, okay, I know that's a tough thing to do, but even to be able to go, well, this is a possibility, like perhaps at a nanotech level, or like you say, by expressing some gene in the body which already has some sort of, produces some protein which does some sort of cleaning, and you modify that perhaps slightly so that it will attack the protein you're trying to clean off.
The way that some of these products are looking at is actually through the immune system, so actually getting the immune system to target and attack and clean up. So using the body's own, tricking the body's system, so basically like vaccinating against alpha-synuclein, so that the body goes in and goes, oh no, we get rid of you. And there's a number of different strategies that people are using that basically it's working around the body's or the cell's system of getting rid of the toxic protein, because what also happens is that the body or cells are very good at getting rid of rubbish. Okay, they've got a really great rubbish disposal system, and for some reason in Parkinson's disease that shuts down. And so and you get this accumulation. That's what this alpha-synuclein aggregate is, this basically the waste
build up.
Yeah, they can't get rid of this alpha-synuclein protein that normally has a really important function, becomes mutated or becomes unable to be discarded, builds up, becomes toxic. So there's other strategies at trying to stimulate the protein clearance pathways and trying to understand exactly, to make it even more difficult, there's different forms of the alpha-synuclein too. So it's like you want to target this bit, but not this one, So, these are, as you say, that's a brilliant idea. You just mop it up. You're exactly on the right track, but then it's,
But the expectation that we as humans can do it in 5 or 10 or 20 years, when the body's probably had spent 300 million years trying to evolve to solve exactly that issue.
And it hasn't. And unfortunately that's, but also, we're a lot older as a population. Usually back Middle Ages, 40 would be a great lifespan. So the brain never dealt with those issues, did they? This is all the issues of aging now that we're getting with a extended lifespan.
And so is there a possibility of replacing and growing new dopamine neurons?
Now you've come to my real passion. That is another way of treating neurological diseases. And so the two that we're interested in is Parkinson's, but also another neurological disorder called Huntington's disease. Less known by people. So Huntington's disease is a genetic disorder. And you're born with an expansion of a certain gene that forms a protein. And that protein actually has a role in every single cell in your body. So we don't want to get rid of that gene, that protein. But for some reason, if you have patients with Huntington's disease have an expanded region of part of this gene that makes this mutant Huntington protein. And again, it goes and forms aggregates, which is what mutant proteins like to do. It selectively kills a population of cells called the striatal neurons in an area of your brain that, again, is involved in organising movement. And Huntington's patients get these sort of what's called chorea movements, dance-like movements, so unable to sort of control their limbs and their bodies, along with a lot of other psychological and other movement issues as well. And again, it's sort of age of onset, similar to Parkinson's disease, so they're both movement disorders. They both have a set group of cells that are selectively targeted, so the dopamine cells for Parkinson's disease and the striatal cells for Huntington's disease. And so what makes that really interesting for people like me is that you can actually target a population of cells for replacement back into the area where those cells have been lost. Wow. So it's not like Alzheimer's disease where you're losing cells in your cortex, which can be a mixture of various different cells. Okay, so it's harder to, okay, we've got some of these cells and some of these types of cells and some of these types of cells, and we need to put a whole mixture back in. Selectively dopamine cells, we can make dopamine cells. Selectively striatal cells, we can make some striatal cells. And when I say by making them, this is where I get really excited. So what we have developed, building on some research that occurred basically a discovery that occurred in 2007, is that we can actually take skin cells from a patient and turn them into brain stem cells.
Amazing.
In a dish, yeah. It's pretty cool.
How?
Yeah. Well, actually, it's actually quite easy. I can say that. It comes on the back of what's known, it's called cell reprogramming. And in 2007, a Japanese scientist, Yamanaka, from the University of Kyoto, demonstrated for the first time that they can take skin cells and they could turn them all the way back to the type of stem cells that's found in an embryo. Okay. So embryonic stem cells, which there's been a lot of discussion in the last 20 years about the use of human embryonic stem cells from no longer required blastocytes from in vitro fertilization and using those for therapy, because an embryonic stem cell is sort of what I like to call the building block of the human body. So an embryonic stem cell can turn into any cell type or tissue type in the body. Just needs to be told what to do.
Is it very basic in its structure? So it hasn't developed the complex.
It's just, no. So it just basically it's just sitting there going, "What do you want me to do?
Right.
What do you want me to be?" And what happens then in normal development is that you get all these different cues that align the stem cells into different regions of the body, the brain, the torso, the legs and the limbs, and then within the brain specifically, the different regions in the brain, and it's all to do with the cues and the signals that it gets from proteins that are floating around. And so you can take an embryonic stem cell and you can expose it to the chemicals and the proteins that it needs to tell it to become a dopamine cell. Okay.
Wow.
But the thing that we were not excited about was that embryonic stem cells, which has always been an issue even with the human embryonic stem cells.
Ethically?
Well, no, ethically, yeah, so we've got some ethical issues about that. But with cell reprogramming, of course, we're taking... I could take your skin cells and I could make you some embryonic. We actually call them induced pluripotent stem cells.
Can you, so I can put them in a jar and just say, look, this is what Dr. Bronwen...
They won't be very alive if they're in a jar. But so there's no ethics around. There's no ethics. There's no issues around that if the person gives consent for their own biopsy of skin cells, right? The issues, of course, around human embryonic stem cells is the use of a fetal tissue that people have various strong opinions about. But the issue around that is that when you're in that stage, that the term induced pluripotent stem cell, they're in this stage that's called pluripotency, which is the ability for them to make anything, okay, and to divide rapidly and continue to divide. That is brilliant, but if you're looking at transplanting cells into a human, what you need to do is you need to make sure that the cells have stopped that pluripotency.
And they're going to be the right type.
The right type, and that you have no more pluripotent cells.
Otherwise, do you get cancer?
You can get tumours. Yeah. So that can form tumours. So we decided that actually we didn't want to go all the way back to an embryonic state. We had no need to. What we wanted to do is we just wanted brain stem cells. We didn't need them to have gone back to a pluripotency state. And then, so it's a bit like going, here's a skin cell which is mature. I'm going to go all the way back to the start, and I'm going to come back here, and then I'm going to go to a mature brain cell. We decided, no, we just want to go from skin cell, brain stem cell.
Cut out the middleman.
Let's just stop going so far. So that's what we developed in 2012. We developed the ability to do that. And we simply put in, so again, using gene delivery systems. So we take the skin cells and we actually use chemically modified messenger RNA, which is the same stuff that we're now using for our COVID vaccines, okay? And RNA is brilliant because it's the safest way of actually inserting genes into a cell, because they don't merge and integrate into the DNA of the person. So there's no issue that you're going to get any genetic mutation. RNA then goes to make protein. So basically, RNA goes into the cell, just starts making the protein that you want it to make. There's no issue of having any genetic mutations occurring. Problem with RNA, though, is that the body senses it and goes, oh, let me destroy you. So we have working with a company in Germany that have developed this special technology where the RNA is hidden from the immune system. So we can put it in, put these two genes in called SOX2 and PAX6, and they're two genes that are first two genes that get turned on.
The very early ones?
The very early ones, they get turned on when the neural tube is being
shared
formed.
with flies and everything. We go right back to the
yeah.
Hox genes. Yeah.
Yeah, and C. elegans, worms and stuff, yes. So they're the two genes that get turned on when you start to form your brain, the neural tube. And so we put those in and feed them for 2 weeks, and we have brain stem cells. And we can do that and just make some generic brain stem cells that can decide for themselves what they want to be. Or we've actually honed it down to being able to put in chemicals and proteins during that phase that makes them become specific stem cells, to become a dopamine cell, for example, or specific stem cells that will only make striatal cells. That means that we can then take those dopamine-specific cells and we can transplant them into the area of the substantia nigra where the cells are lost, and we can replace and generate new mature. So basically put in the babies and then the environment will actually tell them what to do and what to become. So,
and is that something that's near term, to be able to actually generate some of those for someone and then insert them into their brain and see an improvement in their condition with Huntington's?
So with Huntington's disease, we are at the moment looking to patent that technology, and with the vision that that's going to go to clinical trial and hopefully in the near future. So we have demonstrated in rodent models of Huntington's disease that the cells survive and form new mature striatal neurons, which is what we want. Yeah. And that actually the other thing that we do is that we test the rats for their movement function. So the same thing is, because the thing is, like, if you put anything, if I give you a tablet or if I give you a cell transplant, two extremes, and I say this is going to treat your Parkinson's or Huntington's disease in this case, and it's going to take away your symptoms, and it doesn't, and you go, okay, what was the point of that? Well, you've got a whole lot new striatal neurons, so you should be happy. Yeah, but I still have chorea and movement problems. Oh, okay. So what's always important is that we test. We've got a whole series of tests, and I don't like the word test, but there's basically putting the rats into an environment that they would normally be in. So like one of them is an exploring environment. And so we put them into this cylinder so that we can record them, and like a massive big jar, okay. And they can walk around, and we put them in there and we video record them. That's all we do with them. They go in there, they walk around, and of course rats and mice, when they're in somewhere new, they want to explore. They want to find out, okay, where am I? What is this place? And so they if anyone's seen a rat or had a pet rat, usually if you see one in your house, it's running fast. What they do is that they rear up and they put their paws on the wall and they sort of do a bit of walk around, and then they drop back down, and then they have a look, and then they do that again. And so that's really what we do, is we look at their forepaws that they're landing on the wall. Now, rats aren't handed. They don't have a left or a right hand.
No, but they do have hands like us. They
do.
Me.
They have little,
like,
I know, they're very cute, aren't they? Yeah. So they land on the wall, and normally they would land left or right or both, whichever they want, okay? And then they'll move around. And what we do is we record which paw or both paws. Now, if we have an animal of Huntington's disease and we produce the Huntington's lesion on one side of the brain, so they've got one side that's still forming normally, then they will preferentially use the paw that's not affected by the Huntington's. So you'll see them, I only use my right paw to do all my movements. So if we have a therapy that works, they'll start using them both again. Yeah. And that's really strong evidence that your therapy is working.
Are you seeing that?
Yes, we did. We've seen that. Yeah. Back to normal. So, yeah.
Wow.
Yeah.
That's incredible.
Yeah. Yeah, no, we were really excited by it. Yeah, it's really cool. Yeah.
Well, you must be so proud of that, because that's
Yeah.
like that goes back to why you got into science. When you got to be the person.
No,
who
it
says,
is.
"Hey, we
And
can
so,
help
I
with
mean,
this."
it's like, you know, it's been 12 years of really hard work.
Yeah.
And now it's sort of like, it's a bit like, oh my gosh, we've sort of actually got
done
there.
it.
Yeah. So, I mean, we've got a lot of hard work to keep going. We need to get this, this protected. But we need to find investment to set up a company and get things rolling forward and actually get, money for clinical trials. But our aim, particularly with the Huntington's work, is to have a clinical trial in New Zealand, because we have a hugely supportive population. Huntington's Disease Society and all the patients with Huntington's disease have been very supportive of the research that's done in New Zealand, and so they need to be involved and allowed to have this.
And so, without going into commercial sensitive things and details like that, but the process itself, is it a difficult, like if you're going to commercialize it, is it a difficult thing to be able to do to make these neurons and be able to get them so they're in a state they're ready, they won't continue to divide and then put them into someone?
So they don't divide as much as that's the whole beauty of them, is that they don't have that pluripotency. So they don't really divide. We don't have any issues with that. It's a 2-week process.
And what is the delivery mechanism to the brain? How do you get it?
So it's
into
it's called a stereotaxic injection, and it's the same. So Parkinson's patients that are getting deep brain stimulation, they have the little electrode device put into their brain, and it's a bit like having a pacemaker, but in your brain. And so basically, they can have stimulation there that corrects or basically removes some of the tremors and so it's actually little electrical activity that's helping the brain cells function properly, the ones that are left. And that's that's surgery that's done. It used to be done in Australia, but it's now been done here in New Zealand as well at Auckland City Hospital. And what those patients though tend to be quite not end stage, but they're starting to have little effect with their drug therapy. So they have that, and it's basically sounds lovely, but basically it's like you've seen it in the movies, obviously. The cage around the head, right? Okay, and they sit still, and then they just have a big long needle, and they drill a hole, and they big long needle, and they just inject it into the site, and just inject it. So, but that's how it happens. I mean, there is only one way to do this. The same with gene therapy. We're not talking about traditional tablets. This is the type of research that my lab does is not traditional medicines anymore. It's the brain needs something a bit more special to treat some of these disorders.
Is there, and this might be getting way out there, but is there possibility that you could use something like a viral vector to deliver, maybe not just mRNA, but deliver the DNA of the cell, so it became you could actually grow the cell in there. You could effectively give a viral injection or a vector to take it in and then produce in the body.
Yeah, I know what you're saying, because people have tried this. So this goes along an area of work that I spent a whole decade working on before I moved to cell replacement therapy. Well, it was cell replacement therapy, but it was working with the resident stem cell population that you still have in your brain. So in the mid-1990s, the discovery was, so for a very long time we knew that pretty much any other mammal, apart from humans apparently, had a population of resident adult stem cells in their brain. Okay. But of course the human brain was so superior that we wouldn't have such a thing. So it was always dismissed if anyone would suggest it. And then late 1990s, a group at the Salk Institute actually had a very serendipitous experiment where they identified a population of patients that had throat cancer, and they were being given a marker to check the growth of their tumours, which is a marker that gets taken up when a cancer cell's dividing, okay? And so one thing that your stem cells in your brain still do is they still divide, okay? And so they thought, actually, so if they've been given this marker as part of their therapy, and they asked the patients, if you pass away, could we have your brain?
Have a look to see
and
if
we
it's
could
been
have a look at the
picked up
markers in
there.
there. And lo and behold, there's a whole population of cells in two areas of your brain, one called the subventricular zone and one called the in the hippocampus and the dentate gyrus, that still have stem cells. And they are there to make new brain cells. So the subventricular zone, which is the area that I looked at in particular for Huntington's disease, its role is to make new neurons in your nose. Okay, so you have olfactory neurons in your nose, and they die off every 3 to 4 weeks because it's exposed to the environment. And so you have this population of stem cells in this region called the subventricular zone that goes along a pathway known as the rostral migratory stream to your nose and makes new neurons in your nose. And that's just incredible. Yeah. And then the other thing that we discovered, and it's work with Maurice Curtis and Professor Richard Fall, was that some tissue from the Neurological Foundation Brain Bank that we had with Huntington's patients, we could actually see those stem cells moving to the area where the striatal cells have been lost, as if they were trying to go in there and replace. So I built on that work for about 10 years, looking to see what was actually going on, and discovered that, yes, they get redirected from the rostral migratory stream into the area where the cells are dying, as if they're trying to repair the brain. Yeah. Okay. Except, unfortunately, what happens is that because you have an area of cell death and the brain likes to tidy up after itself, it sends in the inflammatory cells from the brain to mop things up. And if things keep on going, if the cell death keeps on going for a long period of time, it becomes inflammation. Okay, it starts to become a chronic inflammation, and it produces quite toxic chemicals. So all these new brain cells were coming in, and the brain's immune system was going, we'll kill you.
Yeah.
Well, they're not directly killed them, but because they were releasing...
the work they were doing, the toxic waste was killing them.
There was a repair process that was trying to happen that couldn't happen. So we spent a long time trying to work out, how could you manipulate this? How could you down-regulate this? And, of course, it's one of those systems where there's about probably hundreds of different factors at play here, and it was just, this is a big soup of stuff, and there's... we looked at guiding them, and that worked, but then you had to mop down the inflammation, but you still wanted those cells to guide, so it was like, we don't want to do this, we want to... So it became quite a difficult process, and we then decided to move into what was actually a much more traditional idea of actually putting the cells back in, because cell transplantation has been around since the 1980s for Parkinson's disease, not clinically as a treatment, but has been looked at with human fetal tissue in the 1980s. There's been a number of clinical trials which had very mixed results because of the difficulty of obtaining the tissue, the way the tissue was then prepared by different groups, how it was. So it was like everyone was doing something slightly different. They'd get different results. No one could tell whether, did it really work, did it not work? And, of course, they were also working on patients that were very end stage. And so I always question, how much success are you going to have at an end stage of a disease? Yeah. And the FDA now has identified that actually we probably need to start looking at earlier patients because you're not likely to be able to do a great repair job or
something that's very
damaged. Yeah. So you're actually shooting yourself in the foot with your therapy before you even start.
Yeah, you're going to end up with failures and bad results because of the task
just can't
is so
fix
enormous.
That. Yeah, we can't fix that. That's impossible. But if we'd got to these people 5 years earlier, that could be a completely different story.
So I guess part of this process is early detection systems. So you can identify early that, hey, you've got Huntington's or Parkinson's before you're perhaps showing those.
So it's
symptoms.
hard. Parkinson's is difficult, as we said, because most patients sporadically produce Parkinson's, and there's no, there's different things that people can look for, but there's no true indicator that you're going to get Parkinson's disease. But even so, if the patient had just come into clinic, they've got the early stage Parkinson's disease, you've still got enough cells there, and if you did some cell replacement therapy, they're at a stage there where you could do a very good regeneration job. Huntington's disease, of course, has the beauty of being a genetic disorder, and so people can choose at any stage to have themselves tested. So it's a 50% heritability. So if you have a parent with Huntington's, you're 50% likely to have Huntington's.
Wow.
So people go and get tested. Some people don't. Obviously there's a huge moral choice factor here. But you could proactively get tested and know that you're going to develop Huntington's and be prepared and ready. I mean, you probably wouldn't transplant before the patient had some symptoms, but it wouldn't be...
You'd be ready to start.
You'd be ready to start earlier on, yeah. So that's sort of the concept that people are now looking at with these therapies, is that we don't wait till the horse is already bolted. It's like, oh, let's fix this. Let's mop up after the flood. Let's not have the flood. Yeah.
So that's Parkinson's and Huntington's. What is Alzheimer's disease?
So Alzheimer's disease is a dementia, and so that has a number of components to it. So you lose the cells around your hippocampus and temporal cortex that are involved in memory formation, so you start to lose memories. You also lose some cortical neurons around processing, and some they quite often get some aphasia as well too, so difficulty recalling words. So it's quite a complicated disease in regards compared to... They're all complicated, don't get me wrong, but it's a lot more multifaceted.
It's not just one cell which is being affected.
And then, of course, you then have the beta amyloid plaques that everyone's very much aware of. So beta amyloid is the toxic protein in Alzheimer's disease. You get this mutated form of beta amyloid. And then you also have tau tangles, which is another protein that tangles up. And there's a huge debate, and the debate's been going on for as long as I've been in neuroscience about which comes first, the beta amyloid or the tau, which is the most toxic, which do we target? And pretty much everyone targets the beta amyloid. That seems to be more predominant. But it is definitely a very multifaceted disease.
It's interesting. My nan, she lived to be 102, almost 103. Yeah, amazing, inspirational woman. Yep. But I do remember a couple of years ago, probably 100 at the time, going to see her, and she had what was being termed, but then was sunset dementia, where in the afternoons when she became tired, sundowning or something.
Yeah, sundowning,
yeah. But I remember going to talk to her one day, and her old memories are strong.
Yeah.
Like, she'd tell me stories which I've heard my whole life of when she was a kid, and they're exactly the same. There's strong memories. But then within the first 15 minutes of seeing her, she might ask me 3 times, how are Renee and those gorgeous kids of yours? And then come back again and come back again. But then what really astounded me was we were there with the kids, and she said, would they like it BISCUIT?
Nothing wrong with their spelling.
No. And the speed of it and the recall. Just astounding. So it affects memory predominantly.
It affects memory. You've got different types of memory. You've got what we call this long-term memory, which is the memories that you're talking about, the stories that they're so ingrained.
Have they grown thick in neurons?
Thick.
Is that what it is?
Yeah, so it's the strength of the connections. Yeah. Okay, so... there's one way. Actually, I really love the movie Inside Out, if your kids have ever seen it.
The kids, I haven't seen it.
Inside Out is fantastic obviously we don't have little emotions with running around like that, but, maybe we do. But, Inside Out is incredible in terms of talking and understanding about how memories form. And I thought for a kids' cartoon movie, it really did a great job. And so you have the things like, what did I eat for breakfast this morning? Okay. Is that really important? And the way I describe it is like, you have a filing cabinet, okay, and what did I have for breakfast this morning? Okay, I had Weet-Bix. Okay, do I need to really remember that? Nah, probably not. Okay, what's the registration number in my car? Okay, do I really need to remember it? Probably. Oh, yeah, probably should put it in a drawer there. Okay, it depends on what type of person you are, right? And then, but, and then it's like, what are my kids' names? Well, that's really important, and we'll put that in the really important drawer, and we use it all the time, and so we always remember, and it gets stronger and stronger and stronger. And then it's also to the memories back into childhood and stuff. They've been around for so long. You strengthen. You tell the story. You remember the story. You think about it. It grows. The connections get really, really strong. But if it, for example, you, for example, your grandmother, for example, might have difficulty remembering the names of someone who she's just met because it's not sitting there.
I have that problem.
Yeah, so do I. What's your name? Because it's just gone into one of the top drawers.
Yeah.
It hasn't been used very often. She may have thought, actually, I don't need to, or her brain's gone, we don't really need to remember that. Yeah. And
doesn't get reinforced.
Doesn't get reinforced.
Yeah.
And your brain is incredible, but it can only actually really hold the information that you're wanting it to retain. There's a whole lot of stuff that's just not important, like what colour socks did I wear yesterday? Well, I couldn't tell you because who cares? Yeah. So there are memories that get solidified and basically they become your long-term memories, and they are the last to go. Whereas you've got your shorter-term memories, your weaker memories, less important everyday bits of information that you don't need to really solidify. You don't think about it more than a passing glance of like, oh yeah, that car was red. Yeah. Okay. And they don't get solidified and they go. Yeah. But then there's always the oddity, my own grandmother also had Alzheimer's disease for a very long time, and she got to the point where she thought my dad was the gardener, and so she'd tell him what things needed to be done in the garden. When she was in a rest home, so whenever he went to visit, and she didn't remember who he was, which is very sad. That's a very sad stage to have your parent get to. But that was that. But she could still tell you about the lolly shop that her parents owned, and that as a kid. Yeah. But she thought the dad was the gardener. So that's the mind just. And it's the pathways that have been lost and stuff, yeah.
So in the way that the technique that you've developed, where you can take a skin cell and turn it into a particular cell type, could we do a similar thing, but for all the different types? Like, could we just get a weekly injection into our brain as we get older and rebuild our brain and avoid?
Probably don't want to go through a weekly anaesthesia, do you?
No, but see, like, is it a possibility? Could we keep regenerating our brain?
I don't know that you want to, though, because you will lose those memories and stuff. There are connections there that need to stay.
But if they were going to die because of old age or because of dementia, if you got a new neural stem cell in this week, could it replace that one, and so that you would keep your memories?
It wouldn't. It would replace it in a different way. So every time so the connections your brain cells make is depending on the experience that it gets when it goes in there.
Yeah.
Okay. And the network. So it's basically, there's a good way of explaining it. You at work, okay, you've got a group of work colleagues and you're all holding hands, okay, and then suddenly a couple of them leave, and then the new person drops in, and you start holding, but they don't know the conversations and the stories, and so then there'll be a new connection and a new set of conversations and stories, and then someone else leaves and someone else comes in, okay, so that cell is going to come in and it's not going to have the knowledge, and it's going to make its own connections and bring its own. Well, it won't have. It'll be pretty new,
so the memory and the story will change.
Things will change. The process that it's meant to do. Because remember, not all cells are involved with memory. Well, a lot of cells aren't involved with memory. They're involved with muscle movement or movement in general, or sensation or things like that, and they need to form the right connection to then be able to do the process correctly. And they might not actually. And you probably don't want to just keep. We don't know what cells die off as we get older. We lose a lot of cells. Most of us, hopefully, retain the majority of our cognitive and mental functioning until we pass away of other causes. So I don't think that would be an idea just for general everyday aging. The things that you can do, though, that will help maintain your cognitive abilities into your later years are just simple things like socialising. That's been shown to have a huge effect on, particularly in delaying dementia. So maintaining your friends groups, going to your clubs or your whatever it is that you do. And also exercise, and we're just talking about going for a walk. So going for a walk with a bunch of friends is great.
Great for your brain.
Okay, but also, going out and talking to people, joining a book club. So exercise and social connection really is the best thing that people can do.
It's interesting you say that again with my grandparents. So Pop died at, I want to say 96 and Nan at 102.
Got strong genes there, aren't there?
But they were both incredibly social people. Like in their late 90s, they would still be having people coming around 2 or 3 times a week socially just to come and see them and hang out. Yeah.
And that's a big thing, I think, with retirement. I mean, I think the generations coming through now, it's going to be interesting because the baby boomers are pretty social people. You know, they go out, they do stuff. Our grandparents, you know, the war babies and stuff like that, they or the pre-war babies, they were used to having an environment where you had to talk to people. You didn't have cell phones, you didn't have computers, you went and talked to people. You walked places and stuff like that. The generation that our kids and stuff is going to be interesting to see their social connectiveness because they have a completely different connection process than we do, you know. As anyone with a teenager knows that it's all about, I'm with my friends, I'm sitting in my bedroom and I'm on my phone on Snapchat and we're all snapping each other. And it's like, right. And the friend is like 100 metres in the house over there and the other one's about 500 metres down the road there. Could you not all just get together in the same room? Well, no, we don't have to. Yeah. And it's like, is that social connection or is that isolation?
It's interesting, isn't it? Because it's a form of social connection, but it misses so much. And on a physical level, it misses things like smell and scent. Yeah, you know, of hanging out with people and being able to pick up on cues and go, hey man, your body, even if you're subconsciously aware of it, like they're not well. Like you can tell, I can tell that you're not well. Here it's like, oh, you just sound like you're texting or we're playing. Whole heap of subtle cues. Yeah,
and I think this is an interesting thing that we've seen with lockdown, with the sudden increase in anxiety and depression that kids are experiencing through school. Apart from the fact that we're much more aware of it, we talk about it now. I don't know that it's been a sudden epidemic increase. I think people are just more, you know, that is a thing. You can now talk about that. I mean, for this country, you can have John Kirwan to thank for that sort of whole thing. It's cool. It's fine. You know, we all can go through periods of time when we need help. But I don't think the teenagers didn't fare well during lockdown because even though we thought, they've got their devices and they can still stay connected to each other. Yeah. I know my 2 boys craved going to see their friends, even though when we weren't in lockdown, I can't get them out to go see their friends. But when you couldn't go and see your friends, what did you want to do? You wanted to go see your friends.
Yeah, and you miss the physical things. Yeah. My two kids are like 7 and 9. They love being physical with me. Like they walk past me and they will just hit me with play because they just want to interact. And one of the most fun things that I can offer to them is, do you guys want to wrestle? Yeah. And they're like, that's the only thing that might beat TV occasionally. Yeah, it'd be like, oh, maybe. Okay, you will wrestle. That physical aspect of their bodies and play. Yeah, missing that.
So it's got a lot to do with, your mental health has a lot to do with social interaction, with getting outside, being with people, getting outside, doing exercise.
And getting those little triggers to get little bursts of, I guess it's dopamine or whatever it is.
It is dopamine,
your friends make you laugh and smile. And, the last 2 hours, instead of just being sitting flat on a device like this, I've been laughing and moving and breathing. And
So, that's probably the only real things that people can actually... there's lots of, do Sudoku every day. Your brain quizzes. It's like,
do it with a friend and it'll probably be better.
Go and have a chat with your mate, even go for a walk. Much better for you. Yeah. So there's a lot of hyped up stuff that, and it's, the brain is an organ. It needs to be exercised. That's what the people say. Yes, keep reading, keep doing your jigsaw puzzles and your crossword puzzles and stuff, but socialisation.
You'll be great at Sudoku, but you'll be a sucky friend.
You're useless as a friend. So, it's definitely in retirement years, post-retirement years, keeping socially active and physically active. Yeah.
Really important stuff. Yeah. What other things are you looking at in the lab? What's coming up, and what areas are you interested in?
Is that not enough?
Well, no, it's just a wealth of amazing information. I want to try and ask as many questions as I can while you're here.
So mainly focused on the cell reprogramming transplantation work. The other thing that we're working on is along in those same lines is cell replacement for spinal cord injury. So I have an incredibly talented research fellow who works for me. she did her PhD with me, and she's continued on. She's actually developed, using the reprogramming technology, she's actually been able to generate a cell type, a very specific cell type in the body called an oligodendrocyte. Try saying that 5 times fast. And so oligodendrocytes, they are involved in wrapping this protein sheath, a bit like, so the cable here has a plastic wrap around it to allow partly safety, but also to allow the electrical signal to run quickly, right? It's the same process in a brain cell, particularly the ones involved in movement, to have really fast. So, you're talking about 300 kilometres an hour. These signals are going up and down these little tracks, right? And so they get wrapped around with this protein called myelin that basically just it's like a myelin sheath. So it's basically just encapsulating them. And the oligodendrocytes make that. That is their job in life. Right. And what happens when you have, so the most traditional spinal cord injury after a car accident and stuff is what's called a crush injury. Okay, and so you don't sever your cord, you're crushing an area, and what happens is that the axons or the tails, long tails of the brain cell that reach out to connect to the other brain cell become crushed, and the myelin gets broken away.
So they can no longer transmit
properly. So Amy has worked out how to generate the precursor cells or the stem cells for these oligodendrocytes, with the aim that we will actually look at transplanting them into a spinal cord model to see if they can remyelinate and therefore help repair. So, and that's work that we're doing at the Spinal Cord Research Unit, and it's part of the Centre for Brain Research.
That's a dream come true.
That would be, wouldn't it? Yeah.
Really.
So, and along those lines too, we're also, one of the other things that spinal cord patients, so we had a symposium last year at Auckland University where we invited, a lot of people involved in spinal cord injury, but also spinal cord people that have a spinal cord injury, and what did they want? What did they want to have? And a lot of them talked about neuropathic pain. So the pain, they still get pain sensation.
Or the phantom pains.
Phantom pains, or whatever you want to call them, but it's called neuropathic pain. So it's like they can have, sometimes I think it just happens. They get this feeling, burning feeling, electrical shock feeling, depends on what the patient experienced.
Quick story, my grandfather, when he's about 30, I think, he chopped the end of his finger off on a homemade saw.
Yeah.
But I'd seen him do this.
Yeah.
Scratching the end of his non-existent finger.
Yeah.
Yeah.
So it's I think it's slightly different to that. I don't know because I've not experienced either of them, but they express it as like, either feeling electric shock or burning or
which you can't then stop or scratch or rub.
You sort of think paralysis. They don't feel anything. Well, it's like, no, that's not correct. They do get these pain feelings, and so how can we actually help prevent that neuropathic pain? And that's also ties into so there's 2 little small projects that we're doing: spinal cord injury for their neuropathic pain, but also multiple sclerosis. So over 50% of patients with multiple sclerosis also experience this neuropathic pain. And pain is really, so I'm not a pain expert, but pain is really difficult to treat because also it's got a whole emotional component as well too. So if you've ever been into hospital and had an injury where you're in pain, or any reason that you're in pain post-surgery, and they'll say to you, on a scale of 0 to 10, what is your pain level? So, your 9 might be my 5 or vice versa, right? And so it's hugely subjective to the person and their emotional state. So it's hard to treat pain. We have opioids, which are not great. Opioids work beautifully, but they're not great long term. And that tends to be really it, okay? And these patients with neuropathic pain, of course, they can't live their lives on opioids.
No.
And then there are a range of other medications, gabapentin, for example, which is actually used to treat epilepsy, but has been shown to have some ability to reduce pain. But none of them are hugely effective. So we're talking about 10 to 20% reduction in pain. So we're looking at some novel compounds to try and see if we can reduce neuropathic pain and spinal cord injury and multiple sclerosis. Not so novel, though, that they can't be. So one thing that I'm really keen on doing as a pharmacologist is finding new uses for drugs that we already use.
Pre-existing.
Yeah. And so so one of the compounds we're looking at is actually a cancer treatment, and it's by its mechanism of action, it should
reduce the pain. Neuropathic pain.
So we're looking at that and seeing whether we can reduce neuropathic pain for.
So the work that Amy and your team is doing to develop spinal cells. So that will reproduce the myelin sheath on crush injuries. Has that had any sort of testing and trialing to see that does it work in terms of restoring function?
No,
along
we haven't got
spine?
there with that yet. That's the dream.
Coming.
That's the dream. So she has just recently she's developed the protocol, which, believe me, it was several years of very hard work. She has now gone off to have a beautiful little boy, and so she's on maternity leave and getting her to try and ignore work. When she comes back, though, then she will be continuing on with that work with some graduate students and another team that works in that same area. And we've got some pretty exciting, combined with a team in pharmacy who do some pretty exciting 3D stuff, and so it's all pretty cool.
Is stuff like that starting to help you now, like the advances in technology, like 3D modelling of proteins? And
no, not 3D modelling. I'm talking about 3D, so hydrogels, so bioprinting, all this stuff, because so we now are moving into, so we've got our cells, now we want to actually put them into 3D little microspheres or scaffolds or whatever, because our brain is not. So when we grow cells in a lab, they're on a plate in a two-dimensional surface, right? And that's how they've always been growing. But they don't
have that scaffolding to allow.
Yeah, well, we are not two-dimensional, are we? So the cells in our brain are not two-dimensional; they're three-dimensional. So we need to have this same three-dimensional capability for the cells to actually function properly. So it's been shown that when you grow the cells on a two-dimension, they don't make their processes quite the same. They can't stretch out in all directions. They don't always respond to cues the same. They don't always signal the same. So we might be getting some false answers out of these cells in two-dimension. And so now the field's really moving towards three-dimensional hydrogels that are biocompatible, that break down. So you could actually, one of the projects that we're working on for Parkinson's disease is to take our dopamine stem cells and transplant them, but also to transplant them in a hydrogel and see if they perform better. Yeah. So there's huge advances, and there's things you can do things with electrical currents and all sorts of stuff now. That's just, but it's also stuff that I as a scientist, you meet people and you go, you do that? Yeah. Well, that is incredible, and it's, I talk to the chemists and the engineers and stuff, and I'm like, I don't know what you're talking about. And then they're talking to me and it's like, okay. And then eventually you come together and you have these very interesting meetings where I talk about small and I'm talking about micrometers and they're talking about nanometers. And it's like, no, we're talking about really small. So, and you get these brilliant collaborations which are starting to happen.
So I was going to ask about, like at the university, because you're in a university setting, is that something that's easily enabled? Like if we need a microfoam gel, can you guys make that for us?
You'd think it would be, right?
Right.
Well, yeah. But, we still like to work in our own little bubbles and our own spaces. And, depending on people's personalities, they may or may not network well. Yeah. They may not, science is traditionally an introverted personality type, right? So, you've got to meet the people to know what they do. And the university's huge, right? And so I had never met these people down in chemistry and engineering because they're actually down on the main campus and I'm up at Grafton. And until we had this sort of serendipitous mutual colleague and we met and talked and it's like, oh my goodness, you can do this, and they're like, oh my goodness, you can do this, and this whole thing just grew out of it. But it's very much by chance, but also, you can put a call out to say, hey, does someone make this? And someone will go, I'm too busy, I don't want to bother, I've got my own stuff to deal with, or someone might go, yeah, hey, cool, that sounds really great, yes, I'd like to collaborate with you, But I always say that collaborations only work if people are friends.
Yeah.
So, it's like you get together, you meet the people, doesn't go anywhere because no one is actually really,
interested.
but also the relationship doesn't work. You meet another group of people and it's like instantly, it's like, yeah, wow, cool, great. Yeah, no, I'd love to work with you. That's great, and you become friends and the friendship and it works around that. So it's not that you work in a university situation so you can just go and take stuff from other people. People are very protective, of course, of what they do.
No, I can imagine they would be. But it sounds like a missed opportunity for the university to somehow not promote that more, because you're doing amazing research here and they're doing amazing research there. If you put them together, the sum of the parts could be fantastic.
Yes, I know. And it's the university tries. Again, people are busy. Academics are busy. We teach as well as run labs. You might have a meeting of let's get all together and see what's, no, I can't come that day. I can't be bothered. Not another meeting. You've got a bunch of free thinkers here that, academics do not what they want to do. Academics, by nature of the job, develop their own day-to-day work. Yeah. And if they choose to come to a meeting, they choose to come to a meeting. You're not going to get everyone in the room. You're not going to get everyone sharing ideas.
You're dealing with artists.
You are, yes. It is. No, it is an art. Yeah. I do say that. And because someone once asked me a couple of years ago, how do you describe science? And I said, it's an art. I said, you have to be creative. Yeah. It's all about creative thinking. Science is science is very, methodical. You have to be very precise with lots of precision. But you have to be creative to think about the next big, exciting idea and think outside the box.
And you need to have that dedication, like artists do. Like, maybe I won't get fed this month, but I just have to do this art. It's like I just have to do this science.
Well,
it's the thing.
99% of the time it doesn't work, and you just have to keep going. And that's what our reality is, And it's always interesting watching new graduate students come in and start a master's or a PhD, and it's like, doesn't work first time. No, this is not an undergraduate teaching lab where we've made it work first time. No, it doesn't work first time. Oh, okay, right.
Probably or second or third or fourth.
Usually we try to keep it going. So there's a definite passion, and I would say not everyone can be a researcher.
Are you finding people coming through? Is it interest in the field that you're actually getting people who like? Neuroscience
is very, very interesting. A lot of women come into neuroscience.
Really?
I just did, I'm not pushing any agenda here, but I went to go and give a talk probably about 10 years ago to the Federation of Academic Women or some group like this, and I just thought, I'll just do a head count on the floor. So we're part of the Centre for Brain Research, and I'm on the floor at the Grafton campus Faculty of Med School, where it's sort of like that. It's a virtual centre, but there's a floor up there that houses as many people as you can on a floor that belong to the centre. And I just did a head count, and with academic staff, technical staff, and graduate students, there was a 60-40 ratio of women to men. Yeah. And I see the same in the courses that I teach too. So a lot of women are coming through into biomedical science and into neuroscience. And we have a large number of growing academic women. Yeah.
I think medical science has always been quite popular with women, hasn't it, in terms of...
No, not really.
Hasn't it? Oh,
no.
I thought that it had been.
No.
Oh, that's really interesting then, that the neuroscience is an interesting field.
Yeah, no, it's been, I think... I'm just thinking back to all of the people, the past predecessors. They've all been men. Yeah.
Right. Okay.
So it's becoming a growing area for women. And as I said, I think biomedical research as well too. They see that. I think that whole ability to help people, that sort of slightly, women like the caring, helping. I'm going to make something to help you get better. I don't want to be a nurse. I want to go into biomedical science and do that now. Yeah.
And the cerebral aspect of it is appealing.
Yeah.
What other, you're saying you teach, what are some of the courses or classes that you teach if someone was to go to the uni?
So I teach third year pharmacology. It's called pharmacology of the brain and body.
And so that's learning about the chemicals or drugs within the brain?
No. So it's teaching students about, so we have a section on the heart, so it's all the heart medications, how do they work? So like we talked about with the medications for Parkinson's disease, how do they all work? So it's talking a bit about what goes wrong when you have a disease, what's the pathology, how do the drugs then come in and change that altered physiology, or how do the drugs then, work to make the symptom or the whatever it is that you're trying to treat in regards to the disease. So the high blood pressure, for example, to lower cholesterol, how does that work? How do those drugs work? So how do your anticoagulants work to stop your blood from clotting? So all of those, basically the broad spectrum of what we call the top 20 blockbuster drugs, which are the ones that are most commonly used. Yeah. And then at graduate level, I teach, starting on Monday, I teach neuropharmacology. So that's a graduate level. So there's the students that are going to go on and do an honours or a master's degree. So that's neuropharmacology. It's basically building on, we take a disease and there's a group of lecturers. I oversee, I'm the course director, I oversee the course, but we have a group of lecturers. Each person's a speciality in, for example, epilepsy research, Alzheimer's research. So they're talking about the field that they do research in, and they talk about the field, what's happening with the latest research, but it's very much designed around how do you do research, how do you design an experiment, stuff like that, so that you're getting them prepared to go into a lab.
To go and research and be effective in there. Yeah,
and then the second semester I teach stem cells and transgenesis, which is a new course for the university. I brought it in probably about 6 years ago now. A lot of universities in America and Australia were teaching about stem cells and also about transgenic animals.
What's that, transgenic?
Transgenic animals, so that's when you make... you alter the genetic. So, for example, you can make a transgenic Parkinson's mouse. So you take the mutated, so this is for the genetic mutations of Parkinson's disease. You can take that genetic mutation and insert it into the mouse. So the mouse has that mutation and is a model of Parkinson's disease.
At like an embryonic stage of the mouse, and then it grows and it will then have that gene to express Parkinson.
Yeah, and you can breed and make more little Parkinson's mice. But also so we have two lecturers from Ag Research come up and talk about the work that they do with basically, some of the milk production that they do. Of course, it's altering the genes to produce increased milk yield, the A2 milk, for example, stuff like that. So
A2 milk, is that an engineered type of milk, or it's a naturally occurring one they're trying to emphasise?
So basically, transgenesis is basically, apart from when you're making a disease model where you're actually purposefully putting in a genetic mutation, genetic engineering, of course, we've been doing it since the dawn of agriculture.
Yeah.
I mean, I hope people are really aware of this when they get excited about genetically engineered crops, that all apples and fruit that we eat have been genetically engineered from another type of apple by just crossbreeding the pollen and all the rest of it, all, putting a branch on a tree and having them. So no, you can select, you can do natural selection as just genetic editing. So, they talk about, so it's talking more about stem cell work that we've just talked about. So a lot more of the more really exciting risque work not risque, you know what I mean? So stuff that, is pushing the boundaries, and we have a lecture, I give a lecture to on the ethics of all of this, about what people need to think about. If you are, choosing, so we talk about cloning, which of course is not permitted, but we talk about cloning, we talk about how you could actually, you can genetically engineer your baby to have blue eyes and brown hair, and is that okay, or is it okay to selectively edit out a gene that might cause a disease in a person? Pre-implantation genetic engineering, stuff like that. So, and a lot of questions that society actually needs to think about, because these technologies are developing and they will be here, and society actually needs to think, what do we feel about this? Yeah,
it's new technology. It's something new that we have to deal with that we haven't had to in the past.
Yeah.
Well, one last question, going right back to sort of the start and the way that you were saying that the treatment for Parkinson's is to put in a protein, the HDGF, GdNF, sorry, which protects those cells.
Yeah.
On a broader spectrum, are there other cells that you could look to do that for as well? Like, could you build a regime of these things that you would give to someone to give a general protection to the brain?
That would be an awful lot of factors because you've got a huge number. So the same, we've done the same type of work for Huntington's disease with another. These are called growth factors that we're talking about. It's another one called brain-derived neurotrophic factor that protects the striatal neurons. BDNF, that's right, exactly. You've got huge numbers. So, yeah, you could, but the cocktail would be pretty ginormous.
Yeah, but in theory you could.
In theory you could, yeah. Practicality, I'm not sure about that one.
Sure.
Yeah.
Cool.
Yeah.
Hey, thank you very, very much.
Yeah, thanks.
I've enjoyed this. It's been a great
that's
session.
Been great.
I think I've learnt so much. But
good.
Thanks.
Yeah.
Okay.
Thank you.
Thanks, Judy. Oh, that was so much fun.
It's my uncut touch cup of tea.
There's some, if you want some coffee, it's still warm, and there's